Let's talk about CRISPR. CRISPR stands for Clustered Regularly Interspace Short Palindromic Repeats. Wow, that's a mouthful. And it took me like 8 times to say that correctly. Basically, these are repeats in bacteria cells that they're going to use to identify viral DNA. Some of the first research that was published on CRISPR was done back in 2012. So this is pretty new stuff. Basically, the CRISPR genes along with the Cas9 proteins enable a bacteria cell to cut and glue DNA in specific locations. Now, there are a lot of intricacies that go along with CRISPR and how it does this. And this system is pretty complicated, but it's really interesting to dive into. The hope with CRISPR is that we can use it in the future to edit the genome of eukaryotic cells, because scientists are hopeful that we can use this system to mutate genes back into functional genes that make proteins that a person may need. Now, I'm only giving a very brief overview of this CRISPR system, but if you're interested more about this topic, I would go give CRISPR Classroom a follow. They know so much more about CRISPR than I do, and I would ask them any questions you have about CRISPR.